So we'll be dealing with the molecular oncology of a brain tumor. The center which are highly equipped with the advanced pathological lab, advanced radiotherapy machine, and a neurophysician, neurosurgeon, as well as a radiation oncologist, high-class physicist. They usually practice the neuro-oncology and then in a most appropriate and a judicial way. So the molecular oncology of a brain tumor is my topic of discussion today. Myself, Major Dr. M. Tubek, Professor, Radiation Oncology, J.K. Cancer Institute, Kanpur, and a UP government institution. So, the molecular oncology of a brain tumor, as the WHO has classified, that's not based on the histology, is no longer the reporting should be done. It should be added with the genetic markers and molecular markers for a brain tumor because of some important fact is considered by WHO in 2016 and added in the classification. How it is useful? It is basically the diagnostic accuracy. First, predict the data of tumor behavior, prognosis of a tumor, and also provide the idea about these targeted therapies. So the diagnostic of use of both histology and molecular test enhance basically the accuracy of a diagnosis. That's why WHO has included in 2016 then the histology should be added with the molecular markers. Sometimes the histology might be very misleading, histology as well as radiological imaging. See the example here, the glioma histologically proven to be appears as a astrocytoma but has a IDH mutation and 1p19 codelations. And another tumor, brain tumor, resembling in histology oligodendroglioma but has a IDH mutant with intact 1p19 codelation. Now what happened here now, in both the situation, molecular type supersedes the phenotype. The actual diagnosis would be in the first case the oligodendroglioma, IDH mutant and 1p19 codelated. In second case, the diagnosis would be the astrocytoma IDH mutant variety. So the criteria for WHO has classified the grading of a tumor is on the basis of 4 factors: the cytological atypia, anaplesia, that is nuclear cytoplasmic ratio, proliferating index, mitotic activity, microvascular proliferation, that is endothelial cell proliferation basically, and the fourth point is the amount of necrosis present inside the tumor. So you see here in the grade 1, out of 4 criteria, there is no criteria is present. Tumor is slow growing, non-malignant, long-term survival, and you see here the right frontal lobe, the clear cut, basically there is no enhancements, no peritumoral edema, tumor is confined, well confined, no contrast enhancement, as well as also you see in the slides. So blue is very less number as compared to the pink, and it has been old dictum to say that the blue means bad and the pink is good. So you can see the tumor is of very low grade, grade 1 almost, but in grade 2 you see tumor is slow growing, it can recur, it can be malignant or non-malignant both. Almost you see here in the cell lines the blue is more basically and the pink are less, and also you see the tumor almost similar situation is there in the different tumor CT scan, but you can see here the slight one portion of a enhancement, but there is almost no peritumoral edema, tumor confined, localized, and no enhancement as such a tumor. So it is graded 2 And in grade 2, only one criteria would be present, that is cytological atypia, out of 4 criteria which has WHO has classified. So the grade 3, out of 4, 2 criteria would be present, mostly the cytological atypia and high mitotic count. You see the tumor are malignant, grade 3, often recur, and a high grade. They consider high grade astrocytoma, high growth malignant gliomas. And you see here the blue is basically, overstudied here, and the pink are very less number. The tumor in the right parietal lobe is have taken a good amount of a contrast. Enhancement is there, peritumoral edema is there, almost aggressive variety. So we grade is the glioma grade 3. The glioma grade 4, if a necrosis present in the center of the tumor, it is almost certain that it's a high grade glioma, which is more common in occurrence. And the tumor is the fastest growing, very aggressive, almost very aggressive. You see here the tumor is having a high contrast, amount of necrosis present, high peritumoral edema, and very high blue in the slide in compared to the pink. So that means the tumor is glioblastoma multiforme, and out of 4 criteria, almost 3 is present. Necrosis is present, high mitotic rate is present, and cytological atypia is present. And sometimes you also get endothelial cell proliferation. So 3, so minimum, and say maybe in a grade 4 you get all 4 criteria of WHO classification of a grading of tumor. You see here this little bit clear picture. You basically see the tumor is crossing, involving corpus callosum, crossing midline, high peritumoral edema in central necrosis, almost very aggressive tumor, and a high grade glioblastoma multiforme. Now, the new nomenclature WHO suggested that the standard terminology should be combined. Histology must be combined with the molecular markers. For example, if it is diffuse astrocytoma, it should be written whether IDH mutant type, or if it is medulloblastoma wingless, whether it is activated, so that the molecular marker should be added. When the more than one genetic determinants are present, the histological name should be followed by the molecular features like the oligodendroglioma, IDH mutant, and 1p19 codeletion. So that gives a complete information of a tumor. What actually you get it, that you can assess the tumor prognosis. Assess the tumor whether the 1p19 codulated means they will respond with a certain type of targeted drugs. And as well as after the completion of a treatment, you can actually presume it is a post-radiotherapy, basically some misinformation in the CT scan, or recurrence. So on the basis of this, you can assess. The new oncologists continued, for tumor lacking genetic mutations, we use the term wild type. Example like glioblastoma IDH wild type. Almost there is no mutation in IDH, so we consider it as a wild type. The term NOS used, not otherwise a specific, basically laboratory those who are lacking a molecular test or a material which is sent for a molecular testing is not sufficient, inconclusive, reports are unconclusive, then we use a term NOS. It's basically used in a different type of tumor as well, like a breast cancer, adenocarcinoma, not otherwise specific. That means you have lacking the molecular test. For the tumor in which the specific genetic alteration is present, use the term positive, like ependyoma, RELA fusion positive means the supratentorial ependyoma, usually the RELA fusion positive and found in the young adults. So what are the basically molecular markers in the brain tumor? is one is IDH mutant, that IDH isocitrate dehydrogenase, 1p19 codylation, chromosomal arm deletion, short arm of 1p, the long of 19q completely deleted. MGMT methyl guanine DNA methyl transferase, TERT telomerase reverse transcriptase, attracts the alpha thalassemia mental retardation X-linked syndrome, TP53 tumor protein 53. So out of all six, the top three is very vital: IDH, 1p19 codulation and MGMT. This gives you a lot of information regarding the tumor management as well as the post-treatment outcome of the disease. So like two types of, if suppose one by one we consider the IDH mutant, the IDH has basically two types of variety, IDH1 and an IDH2. The IDH1 mutation usually happen in the codon 132, which substitutes glycine to the arginine and lead to the abnormal production of 2-hydroglutarate, causing histone and DNA methylation, therefore cause tumorigenesis. The IDH2 mutation usually occur in the codon 172 associated with 2-hydroxyglutaric aciduria. Also, this kind of variety of patients present with seizures, muscle tone, cerebral damage. So you can, if you have IDH2 mutation, you can predict, you can start the anti-seizure drugs, because you know that these people are likely bound to have a seizures and the cerebral damage. So significance IDH being the most important diagnostic marker in differentiating between the glioma and gliosis, positive in astrocytoma and oligodendroma, as well as in 10% cases of GBM, IDH positive is found. So let us be very clear about the glioma and gliosis. Gliosis usually because of brain injury, brain hemorrhage, strokes. The gliosis is something like a fibrosis of a soft tissue, and a fibrosis of a brain is a gliosis. And a glioma, the gliosis will be present, but this IDH can differentiate whether it is of a gliosis or whether it is because of glioma. So radiological significance of IDH. IDH mutant glioma tend to recur. IDH mutant tumors rarely degenerate into glioblastoma. IDH mutant tumor peritumoral edema is less likely. Well-defined border. IDH mutant glioma generally lack contrast enhancement that you will see. The IDH mutant, IDH positive, the well-localized tumor not crossing the midline, very well confined. Borders are sharp, very less peritumoral edema. But in the IDH negative, you see the tumor is crossing midline, aggressive, high peritumoral edema. So that then you can predict the outcome of a disease. The IDH negative is having a poor prognosis. So that's the second marker, the 1p19 codylation, the complete deletion of short arm 1p and a long of 19q. If it is present, and how we assess by either the FISH, the fluorescent in situ hybridization, polymerase chain reaction. These are the tests we perform for this marker, and the significance of this test is the definite diagnosis of grade 2 and grade 3 glioma can be made because of this. The 1p19 codulated if suggests the better outcome of a disease, improved survival, and positive predictive response to the chemotherapy, that is alkalizing agent like the temozolomide, PCV regime, as well as radiotherapy, both. You can see, go through that important research article regarding this, the prediction of 1p19 correlation using the preoperative multi-parametric magnetic resonance imaging. So you see here in the 1p19 codulated and not codulated negative. So negative is having a large irregular tumor crossing almost midline as a pressure effect, and then those tumor who are having a 1p19 codulated having a well-confined, localized tumor, less aggressive variety, and we can expect a better outcome, outcome with the temozolomide, better outcome with the radiotherapy, and that you can see in the larger image the same picture. The third marker is very important, MGMT, methylguanine methyltransferase gene. MGMT gene encodes a DNA repair enzyme depending on its promoter status. If a promoter region of MGMT gene is unmethylated, the gene will be active. If the gene is active, making the tumor resistant to the chemotherapy by alkalizing agent. But if a promoter region of MGMT is methylated, hypermethylated, gene will be silenced, and that these people will respond to the temozolomide drug and will have a better outcome. The third marker is TART, that is telomerase reverse transcriptase. TART promotes the mutation. It usually occurs in the GBM and a high-grade oligodendroglioma. Significance: it predicts the poor survival. If it is positive, its poor survival predicts the radiotherapy-resistant glioma. The radiological significance of TART: majority of morphological features are not significantly different. The TART mutant tumor had a high blood-brain barrier dysfunction. The ATRAX alpha thalassemia mental retardation syndrome is inversely proportional to the TART. So these two, if ATRAX positive is there, it's almost a welcome sign that tumor is basically, if it is positive, that means because it works basically interlinked with the telomerase, and a telomerase work, we all know that it repairs the DNA before it goes to the next cycle for a mitosis. So if it is intact, so it helps in the DNA repair. So ATRAC mutations are inversely proportional to TART mutation. So the ATRAX positive improve the survival of the crunch point. The glioma marker p53 tumor protein is gene located in the short arm of chromosome 17. Identification of this gene, loss of p53 gene is directly related to DNA damage, hypoxia, oncogene activation, microtubule disruption, oxidative damage. These turn contribute to CNS tumor pathogenesis, especially in medulloblastoma, glioblastoma, IDH mutant testocytoma. So, p53 is a favorable gene, but if it is mutant, the following features will be observed and the prognosis will be poor. So, you can see in the case 1 and the case 2, the p53 gene, that the one is positive, wild type, there is no mutation, having a good radiological picture, seems to be appearing a better outcome with the treatment. But in the second image, you see the p53 mutation is there, an aggressive variety tumor, high peritumoral edema, large size, outcome is poor. Other molecular markers is not basically so much is being studied till now, just restricted to the academic purpose. So with this, we end up this molecular oncology section of a brain tumor, that is section 1. I'll be coming with the section 2 of a radiotherapy planning in the molecular brain tumor. Thank you very much.